As the best electron acceptor, oxygen plays a crucial role in biological systems. With this protocol, you can image the oxygen distribution in 2D using an optode.
In addition to its high spatial and temporal resolution, this method does not require an additional reference dye, is extremely robust, and allows the acquisition of a structural image.
This method can be applied to various areas of research in which oxygen plays a key role, including ecology, medical research, bioprinting, and pressure-sensitive painting.
The optode fabrication can be challenging, as the amount of cocktail added to the support foil and the knife coating device drag speed may require optimization in practice.
With this video contribution, we want to make this powerful technique accessible to researchers from other fields of study.
To prepare a planar oxygen optode, first use a film of water or 70% ethanol to fix a clean, dust-free PET foil on a cleaned glass plate. Place a clean 120 micrometer knife coating device onto the foil and use a glass pipette to apply a line of sensor cocktail in front of the device. Next, drag the knife coating device slowly and uniformly over the PET foil to spread the cocktail evenly. Then dry the finished planar oxygen-sensitive optode in ambient air for 1 hour before drying overnight in a heated cabinet at 50 to 60 degrees Celsius. By the next morning, an approximately 12 micrometer thick layer will have been obtained. Store the optode protected from light until further use. To prepare a rhizo sandwich chamber, use light curable acrylic-based instant adhesive to glue microscope slides along the edges.